What's up, baby bio penguins? So, slight technology issues. So my laptop currently is low on battery. So if for some reason I lose my charge on my computer, then I will grab my husband's computer, which I've already got the PowerPoint all loaded up. So welcome. What's up, baby bio penguins? Today we're going to look at my Unit 4B. My students have a unit exam tomorrow. Over 4B for me. And so my 4B is for most of y'all. Oh no, you can't hear me. Uh-oh. Is my earbud not working? Can you hear me now? Can you hear me now? Okay, I'm gonna come off. Oh wait, you can hear me now. Okay, awesome. So I guess that earbud was just not working. So let me try that again. My husband's computer is on just in case because of the fact that my battery is low and I apparently don't know where my charger is to my laptop. So tonight we're going to do 4B. 4B for my students is, for most people, Unit 6. So it's going to be like replication, transcription, translation, the operons, and then all the DNA technology. So what we've done in the past is I just go over some different information that I have. You're more than welcome to pop a little question down the little question box, and I can answer your questions as we go along. I also have the phone in front of me so I can actually see your comments and your questions as you type them into the chat. Just let me know if there's anything you need in terms of Unit 4B, because we're focusing tonight on 4B. My students have a test tomorrow, so I'm just going live so that we can kind of do a quick review over the big concepts to make sure that we are prepared for our test that my students have tomorrow. So without further ado, let me flip the camera. So as I said, Unit 4B is mostly Unit 6, so I'm going to do a quick thing on replication, although it's not my student's test, just because replication usually is all with the molecular biology. We'll do transcription translation, we'll do operons, and then we'll do our DNA technology. So real quick, with DNA replication. DNA replication is going to take place in the nucleus. And so with replication, the job of that is that we're going to start with a DNA strand and we're going to make another DNA strand. So we're going to replicate our DNA. And so the big thing about it is that DNA is anti-parallel. And because DNA is anti-parallel, it causes a lot of issues because of the fact that we need to have two different strands. And so there's a leading strand and there's a lagging strand. Your leading strand is going to move continuously into your replication fork. So as your DNA strand comes apart, using the enzyme helicase is what's going to break those hydrogen bonds to unzip or open up that double helix. So we're going to break those bonds to pull apart the DNA helix, right? So then our leading strand is going to be the one that's going to be made 5' to 3'. Okay. And so it's 5'. I will, of course, post a live later. The 5' end is where our phosphate group is. And so we're going to move from the phosphate group towards our hydroxyl group, going 5', 3' into our replication fork. That's going to be our leading strand. So as I continue to open up my replication fork, as I continue to split those two DNA strands, I'm going to continue to make that copy continuously into the replication fork. So it's called the leading strand. The opposite strand is called your lagging strand because it's only going to be able to work discontinuously. As I open to this point, I can make it a little bit. And as I open up this point, I can make a little bit more. So it's going to keep jumping back and going back. So that creates these little things called Okazaki fragments. Okazaki fragments are just these short little fragments of DNA. And so we'll use the enzyme DNA ligase afterward to seal those little fragments together. So let's look at a little picture just to make sure we got this. Okay. Something I forgot to mention. We always have an RNA primer. DNA polymerase is unable to build off of nothing. So you're always going to have an RNA primer to start. Your primer is going to start, and it's going to provide a opened 3' end for the DNA polymerase, the enzyme that allows for DNA replication to build off of, for it to be able to then move. As I said before, we've got two different strands. Our leading strand is going to be moving continuously into the replication fork. So as this strand unzips, as we pull apart this strand, and I realize I used my other hand, right? So as we unzip, as we open this up, this leading strand is going to be moving towards that replication fork. Versus our lagging strand is going to be moving discontinuously. As I move a little bit, it comes back. Okay, so that's our quick little look at replication. Replication has also been studied in probably the 4A video that I did, just because I knew that my students did it in 4A. I split it up, but most teachers don't. So depending on your teacher, you may not. So moving into central dogma. So the central dogma is the way that gene expression takes place. So we start out with DNA, and we're going to read the DNA strand. Then we're going to go through transcription to transcribe the DNA to make an mRNA strand. That mRNA is then going to be translated to make a polypeptide. Now we say polypeptide, not protein, because not all proteins are a single polypeptide. So some proteins are made up of multiple polypeptides. If you remember back to Unit 1, there were those different levels of folding with proteins. And the quaternary structure dealt with when you had more than one polypeptide. And so we often say with the central dogma, it's DNA to RNA to polypeptides. Now, there's some things that violate this central dogma. So an example of something that violates the central dogma is HIV. HIV is an RNA virus, which means its genome, its genetic material, is RNA. And so what it does is it takes that RNA and it uses an enzyme called reverse transcriptase. And reverse transcriptase is going to allow your RNA to be a template, and it makes a DNA strand from that RNA. And so then it inserts itself into your white blood cells. And so we've gone from RNA to DNA. So it violates that central dogma. And there's an FRQ question. Oh gosh, I do not remember the year off the top of my head. But in that question, you go through transcription and translation, and then it asks you about a virus that will violate the central dogma. So again, looking at a picture, our central dogma is starting with DNA. When we go into transcription, we'll transcribe that DNA into RNA. We'll then translate that RNA into polypeptide, which is just our string of amino acids that we can then use to make, of course, our proteins. So moving on. So transcription is going to take place depending on whether we're in a prokaryote or a eukaryote. Prokaryotes are going to have transcription and translation take place simultaneously. That means they're going to happen at the exact same time. So as soon as you start transcribing, you start translating. And the reason why is because a prokaryote doesn't have anything that separates the DNA from the ribosomes. So in a eukaryotic cell, we have a nucleus, right? We have this nuclear envelope, this thing that surrounds our DNA. And so traditionally, in transcription for a eukaryote, we're going to go through the transcription process. We're going to read the DNA. We'll make our RNA within that nucleus, okay? And then after we make that mRNA, then it's going to leave the nucleus for translation to then find the ribosomes for that translation to take place. So in a prokaryote, it happens at the same time because there's nothing separating them. Versus in a eukaryote, transcription and translation are two separate processes. Because of the fact that there are two separate processes, we're going to see different things take place in a eukaryote after we transcribe, but before we translate. That's going to be called post-transcriptional modification. It's different ways that I can modify to regulate the translation of it and regulate that gene expression. So let's look at transcription here. So transcription is going to take place using the enzyme RNA polymerase. So RNA polymerase is just like DNA polymerase in terms of that is an enzyme that is going to read the DNA to synthesize something. And the enzymes always say what they do, right? So RNA polymerase is going to make an RNA polymer, right? So the way that the place where it starts is called this promoter. So we're going to see that our RNA polymerase is going to bind to the promoter. Okay, so once it binds to the promoter, it quickly kind of pries apart the DNA strand, and then it's going to go through complementary base pairing. The difference in transcription versus replication is that now that I'm making RNA, instead of putting a thymine, I'm going to put a uracil. So anytime that I'm base pairing to an adenine, there's going to be a uracil complementary base pair to it. Versus whenever there's a cytosine, there's always a guanine. That's the same thing that you see in DNA. And if you see a thymine in your DNA, then you'll have an adenine, just like what we saw with DNA. So the big difference that we see whenever we are transcribing, is that anytime that you would have a T, we're going to have a U. So a test-taking strategy here would be if you know that you're looking for RNA and you see that an answer choice has a bunch of thymines in it, you know to eliminate that one. That one's not an answer choice because RNA DNA does not have thymine, it has uracil. Okay, so the RNA polymerase is going to bind to our promoter region, and it's going to pry apart the DNA strand so it can then start reading it. Okay, so here you see we've pried it apart, and it's going to start moving in. After we've moved past that region, the two strands come back together. There's nothing really needing to keep them apart. We just need them to be apart just so we can complementary base pair. So you can see here we have that mRNA coming out until we reach, in prokaryotes, we have a terminator sequence, so it falls off. And then in eukaryotes, it just kind of goes through this polyadenylation sequence. Polyadenylation just means there's a whole bunch of adenines. And so once it falls through that polyadenylation sequence, it falls off of the DNA, and now it's ready to roll. Translation is going to take place in the ribosomes. So you learned about the ribosomes in Unit 2. We talked about all of your different organelles, right? And all of our ribosomes are technically free ribosomes. And so those free ribosomes can bind to a nucleus, specifically the ER, if need be. But so the big thing about translation in the ribosomes is that there's two big subunits. There's two subunits to it. You have a large subunit and a small subunit. So I always like to think of it like a SpongeBob, those Krabby Burgers, what are they called? I cannot think. Anyways, but they're little, you know, the burgers I'm talking about. Anyways, so the top part of it is a big bun, right? And so that's going to be your large subunit. The large subunit is going to have 3 binding sites on it. You have the A site. At the A site, this is where we're going to add the new amino acid. Okay, so we have a tRNA. Thank you, Krabby Patty. So we have a tRNA that's going to enter, and that tRNA is going to have an amino acid attached to it. So it's going to enter at the A site. At the P site is where we're going to have our growing polypeptide. Okay, so we're going to have a tRNA that's sitting there that has this really long polypeptide chain attached to it. And then the E site is where we're going to have an empty tRNA exit. So overall, what's going to happen is that we're going to add a new amino acid in at that A site. The tRNA that complementary base pairs, the tRNA that has the anti-codon that pairs up with the mRNA's codons, is going to enter at the A site. We're then going to create a bond between that polypeptide that's on this one, okay, and that new amino acid. So we form a new peptide. We form a peptide bond, right? And then we break the bond here. And then when that happens, we then translocate and we shift everything over. So what was at the P site, now, I don't have enough fingers. What was at the P site now moves to the E site and exits because it's now empty. And what was at the A site now has that polypeptide. So now it moves to the P site, and now we're ready to take a new amino acid in at that A site. Okay, and it's going to keep doing this as it keeps moving forward. So translation is going to start at something called a start codon. And so how do we know that we got to the start codon? So the small subunit binds to your mRNA. And so you're going to keep moving on the the mRNA. It's going to keep moving until we reach the start codon. Start codon is AUG. The way to remember that is that school traditionally starts for a lot of people in August. So AUG is the start of school. And then I always tell my students that I'm an only child, so I'm the center of the universe, and my birthday is August. So of course the world revolves around me. So of course start codon is AUG. Doesn't really work for everyone, but hey, I like to say it. So start codon is when we'll have the whole subunits come together, large subunit, small subunit will come together, and that's when our first tRNA comes in. And then the elongation phase is what I was mentioning before, where it just kind of new amino acid comes in, polypeptide on the P site, and then of course exit on the E site. So let's look at a picture just to make sure that we're completely understanding this. And I do see that there was a question in the question box, so I'll pop down in just a moment and answer those questions. So here we see we have our small subunit. Our small subunit is going to be attached to the mRNA. It's then going to allow us to, when we get to the start codon, that AUG, that tRNA, that complementary base pairs, is going to pair up, and it's going to have methionine on it, okay? So next, we're going to see that we're going to have that A site, right? The A site is where the new amino acid is going to come in. Notice that is it going to zoom? Okay. Notice that it complementary base pairs. So we have a UUC. So the AAG pairs up with it. And it's bringing in that phenylalanine. That methionine moves over into the P site. And of course, our E site is currently empty. And so what's going to end up happening is that we're going to break that bond that we have right here. So the methionine is now going to be bonded to the phenylalanine. The whole thing's going to shift over. And so now I have that growing polypeptide on the P site, break that bond, and now it's going to be attached to, it's because it's at the bottom of the screen, attach the arginine, and then the whole thing will shift over. Okay. So there was 2 questions in the box. Let's see if we can answer those real fast. Make sure. I'm not answering that question right now. Okay, so that is what we see taking place there. And apparently I did not put a post-transcriptional modification in here. So after transcription, before translation, we will modify the RNA. And so 3 things will happen. Number 1, we're going to add a 5' cap. So we add this cap to the front of it. And by adding that cap, it provides a location for the small subunit to bind and know where to start reading. It also facilitates the export of the mRNA from the nucleus out into the cytoplasm. The second thing that's going to happen is we're going to add a poly-A tail. Poly-A meaning a bunch of adenines. We add a whole bunch of adenines to the end, or the 3' end of the mRNA. This is going to prolong the life of the mRNA. There are hydrolytic enzymes that are in the cytoplasm. And so those enzymes are going to constantly kind of deteriorate and break down the mRNA. And so the longer poly-A tail that you have, the longer the mRNA is going to hang out in that in the cytoplasm to be able to be translated. So we'll be able to make more proteins from that mRNA. And then the third thing that's going to happen is going to be splicing. Okay. And so we're going to cut out the introns and put together or splice together the exons. So the way I help my students remember that is that we're going to keep the exciting, expressed exons that all start with that EX. So exciting, expressed exons are kept, and we're going to cut out those introns that are in between and in the way, and I don't really want them. Right. And this leads us into that alternative gene splicing, or RNA splicing, or alternative gene regulation. This has to do with that we can use the same mRNA and then make multiple mRNAs to leave to be able to make proteins. Okay. So your pre-mRNA and then the actual mRNA that leaves. You can denote which ones are exons and which ones are introns, and they can cut out extra different parts. So I make my students do this whole thing with A Whole New World lyrics, and they create different phrases from it. And it's amazing because with a classroom of 20 kids, we maybe have one duplicate. And that just shows you that within, your body, your mRNA can make tons and tons and tons of different mRNA strands from that, which is why we don't have to have as much DNA as we have proteins in our body. So, on to mutations. So sometimes an error happens, right? And this error is something that's in your DNA. So a mutation is not something that's happening in the RNA. Okay, so yeah, you could have a slight error, but that's not really what we're calling a mutation. And I do see there was a question in the chat. It says, what happens if introns are left in? If the intron is left in, it could cause something called a frame shift mutation, which we'll talk about in just a moment, because it could alter the reading frame. Traditionally, they're just junk. There's nothing actually in the introns. It's usually coding for rRNA or siRNA or miRNA, other things that are not actually part of the mRNA that's going to be expressed. And so we usually won't see much happening with that. But yeah, if the introns are left in, you could basically be wasting time and resources, or it could cause frame shift mutation, depending on where there's a stop codon. Okay, so mutations are going to take place in your DNA. So there's some type of change that happened in the DNA. And because of the change in the DNA, you're going to see a change in the RNA, which of course leads to a change in your polypeptide sequence. So there's 3 types of point mutations. And a point mutation is something that's going to take place at one point or one nucleotide. So here we see the 3 different ones we could have. Okay. So you could have a silent mutation. So in a silent mutation, you see that here I have a uracil, and it's been substituted with a, how is that an original DNA? This is a bad diagram, y'all. Anyways, so instead of there being a uracil or a thymine, it's got a cytosine there, which of course would be a problem, because now we've got a different amino. Oh wait, I'm sorry. I'm definitely in limbo. So instead of the uracil, we have a cytosine, which could potentially be a problem, but because of the fact that it codes for the exact same amino acid, so see this codes for leucine, and here we see that it codes for leucine. So since I have the exact same amino acid, this is called a silent mutation. You have no clue that a mutation took place because it still makes the exact same protein. It's still functional because it's not actually a change in that protein. The only way to know that there's been a change or that there's been a mutation is if you actually look at the sequences. So the next one we have is a missense mutation. In a missense mutation, when I switch out the uracil and the guanine, instead of it being leucine, now it's going to be valine, okay? So instead of one amino acid, it's a different amino acid. This could cause no problem at all. Like maybe it's not in the active site. Maybe it's not in a part of it that allows for the folding of that protein. So it could have no effect. But it also could have a substantial effect if it's part of the active site, if it's part of the way that it folds, right? So we talked before about that there are polar and non-polar side chains, right? A polar side chain is going to fold outwards towards the polar hydrophilic environment, right? Versus our non-polar side chains will fold inward. So if you substitute a non-polar R group for a polar R group in terms of your amino acids, it could completely change the folding of that polypeptide. If you change the structure of a polypeptide, you change the function of it. So it could have detrimental effects. So missense could go either way. If you have a question like that on your exam, you probably want to use your best guess, give a prediction, or use the prompt. It might give you some information as to what would potentially happen. And the last type of point mutation we have, in terms of substitution, is that instead of a uracil for the second one, we have a adenine, which codes for a stop codon, which is considered a nonsense mutation. So it's considered nonsense because the amino acids literally stop. They're truncated. You have a very short polypeptide. Again, could have no effect if it's at the very end. Also, could have no effect if it's not in an exon or if it's before the... What did I just do? Anyways, so it could have no effect at all. But again, if it's in the active site, if it's in part of the way that it folds, it may not fold correctly, which could lead to major problems because it is a shortened, truncated amino acid. Okay. Another point mutation we can have would be insertions or deletions. Insertions or deletions are detrimental. They are bad. They are really bad to get an insertion or deletion of a single nucleotide. So the reason why that's bad is because codons are read in groups of 3, right? So we find our AUG, that's start codon, and we look at every group of 3 letters going after that. So if you insert or delete one nucleotide, that group of 3 is now off center. And so you're not reading the right 3 letters, so you're not attaching the right amino acids, which can cause major problems to your polypeptide. So you have to think, and they might ask you, well, you have a mutation, right? You see this frameshift mutation. Where is it going to be worse at? Would it be worse in a intron? Would it be worse after the start codon? Or would it be worse right before the stop codon, but at the end? Well, it's going to be worse right after that start codon because it causes every single one to change. It's not going to have any effect if it's in an intron, and it's going to have a very small effect if it's near the end of that polypeptide. So it kind of has to do with a little bit of information that you're going to get from your prompt. So I see that there's a couple questions in this chat. Do nonsense mutations always code a premature stop codon? Yes, because of the fact that our amino acids should keep going. We should still be getting more amino acids, but we've substituted it for a stop codon, which means that we have now stopped the amino acid. We've stopped the translation, so it will always make a premature stop codon. I see another question. A nonsense mutation, specifically a mutation that changes the codon to a stop codon. Yes, a nonsense is always going to make it a stop codon. So silent mutation is going to allow for the exact same amino acid. The missense mutation will code for a different amino acid. And the nonsense mutation will code for a stop codon. So there's your 3 there. And then frameshift mutation is, of course, going to shift if you insert or delete. So something that isn't going to cause big problems in terms of insertion or deletion is if you insert in a group of 3. So if you add 3 nucleotides in, or if you cut out 3 nucleotides, it's not going to cause as big of a problem because only those 3 and then you're back on track. Okay. So that's another thing to look for in terms of the questions. Okay. So, the thing we all hate, operons. So operons are made up of 3 different major components, right? So you have your promoter, and we've talked about before that the promoter is where the RNA polymerase is going to bind. We also have an operator. So within that promoter region, you see here we have an operator. The job of the operator is to bind to the repressor. And then you have all the genes that it kind of expresses, right? So it's going to read all these genes. Now, important note, operons are only in prokaryotes. You will not see an operon in a eukaryote. So there's 2 main types of operons we have. We have repressible operons and we have inducible operons. Repressible operons start out being on and they get turned off. Okay. And so the way that they get turned off is because of the fact that there is a repressor that is active, and that active repressor is binding to the operator, which is inhibiting RNA polymerase from binding. Okay. So of course the operon will be off. An inducible operon is going to start out being off. And in the presence of a certain chemical, it's going to cause the repressor to become inactive. And so since your repressor is inactive and can't bind to your operator, that is going to cause your operon to be turned on. So inducible operon gets turned on. So it starts out being off and gets turned on. So I usually will turn the lights on and off on my students in the classroom. So that's why I was turning on and off on our little live stream. So the trip operon is an example of a repressible operon. And so what was going to happen with the repressible is that we're synthesizing something. We're making something. And so if that something I'm making is already present, I already have a bunch of it, why am I going to waste resources making something I already have? And so what's going to happen is that product is going to come back and it's going to bind to your repressor to cause your repressor to bind to your operator. So it's like a big relay effect almost, right? So let's look at a picture of it to make sure that we understand what's happening. So here is our trip operon in its normal format, like when it's first made, right? So because of the fact that my repressor, where's my repressor? No, this is the repressor. Anyways, because the repressor is inactive, it's not binding to the operator. So RNA polymerase can bind, and it can transcribe these genes, making my mRNA, which will then make my protein, which will allow me to make the amino acid tryptophan. Okay. And so if tryptophan is present in high concentration, so I have a high amount of tryptophan is going to bind to my repressor. And by binding to my repressor, the pressor now binds to the operator. So RNA polymerase is unable to bind to the promoter region because there's this big thing blocking it, right? And so because of that, now I'm no longer going to be making tryptophan. Well, that's fine. I have enough tryptophan. I don't need any tryptophan. So this is a way to have negative feedback, right? Instead of wasting resources, the product comes back and inhibits the process to inhibit, so we will not waste resources or energy. So a repressible operon is an operon turned on by repressor. You have it backwards, darling. A repressible operon is going to get repressed. It gets turned off. An inducible operon is going to get turned on. So horrible way to think of it. But think about like induction of labor. So if there is a woman who needs to have their baby, well, actually it doesn't have to be a woman. If there is a individual who's having a baby, they can oftentimes induce labor by giving the medication to start contractions. So that's what I'm trying to say. Like inducible operon is going to start something. It's going to take something that's not on and turn it on and make it go. Okay, so we're, I think, up to date. I think the student is perfectly fine now. Okay. And then there's a question in the box. Let's see. Brain fart. If operons are only for prokaryotes, how is gene expression regulated in eukaryotes? Let me come back to this in a second. Okay. I don't have a PowerPoint for it, but I'll talk about it in just a moment. Okay, so the other operon we have was the inducible operon. So with our lac operon, this is traditionally going to be turned on in the presence of something, okay? And so in terms of our lac operon, it's going to get turned on in the presence of lactose. So the inducible operon is going to make the materials to break down lactose. If lactose isn't present, I don't want to make the stuff that's going to break down lactose because there isn't any lactose there, okay? And so what's going to end up happening is when lactose is present, the lactose is going to come and bind to the repressor to turn the repressor off, which will allow it to no longer bind to the operator. So now RNA polymerase can read and make your genes to then, not make the genes, but make the mRNA to make the polypeptide to be able to break down that lactose. So let's look at our picture, make sure we are completely on it, okay? So traditionally, it's going to be off. So here we see our repressor is bound to my operator, and I know it's not as pretty as the other one. Inhibiting RNA polymerase from binding, so we're not going to read anything. But whenever there is a presence of lactose, lactose is going to bind to my repressor, turning my repressor off so it no longer binds. RNA polymerase can now bind to then transcribe these genes so that I can break down my lactose because now my lactose is present. So I hope that was helpful. And so somebody asked about eukaryotes. So with eukaryotes, we have our genes on different chromosomes. And so like if we're trying to make some specific material in our cells. It's going to be on different chromosomes. So you're going to have multiple promoters, multiple RNA polymerases, and then, of course, if you're trying to stop it, multiple repressors. And so oftentimes, in terms of activation, we have things called transcription factors. Transcription factors are going to bind to that promoter region, which stimulates the RNA polymerase to bind. And so that allows for increasing of gene expression. We also can modify gene expression by acetylating the histones. We can methylate the DNA. That's how you make bar bodies, is that you methylate that X chromosome, which causes it to condense and become like a little ball of genetic material that you see in the wall of the nucleus. Not really the wall of the nucleus, but the plasma membrane of the nucleus. So that's how you can kind of do it with eukaryotes, but it does take multiple promoters and multiple regulatory sequences. Someone says in the chat, is there a place where the notes can be found? Yes, all the notes are already posted onto apbiopenguins.weebly.com. That's where all of my stuff is. So if you go under there and you go to the Jones class Instagram, since this is supposed to be for my students to review for their test they have tomorrow, that's where you would find it. Okay, so biotechnology. So the last thing we have to talk about is biotech, which is an entire whole section. And oh no, my laptop is dying. So we might be switching to my husband's laptop in just a second, because I know there is for responses if you wanted to go over those. So the first thing we have is PCR. PCR is polymerase chain reaction. And so basically it's a chain reaction. You put all this stuff into the apparatus. You put your primers in there. You put a heat-resistant DNA polymerase in there. You put in free nucleotides. And this thing literally will just go over and over and over and over and over and over in this thermocycler by heating up, which will denature your DNA, basically pulling the strands apart, cooling down by allowing the primer to attach, and then making this polymerase and basically allowing the primer to then build a new strand. So let's look at a picture. So here we see, of course, that DNA within our PCR apparatus. You're going to have our primers, our nucleotides, buffer, and all this fun stuff. And so what's going to happen is it's literally going to make multiple copies. So after one round of division, we can make 2 copies. After 2 rounds of division, we make 4 copies. After, I don't know what number I'm on, but 3 rounds, we're on 8, 16, 32, and it keeps increasing over time. So I think like after 30 rounds, we're at like 1 million or 1 billion nucleotides. It exponentially grows. And so the purpose of this is so that I can make more copies of my DNA. So sometimes there's only a small sample of DNA, you need more of it in order to test it. And then sometimes you just need more in order to be able to see it. So especially if you're running something called a gel electrophoresis, a gel electrophoresis job is to separate DNA or RNA based on size and charge. And so we use a gel that has these little pores in it. And so then you turn on electricity. And so by turning on the electricity, electrical current runs through it. There's like a buffer that helps with the electrical current. And so it pulls the material from one pole, the negative pole traditionally, to a positive pole. And so that's going to allow you to separate something. And so if I only have enough DNA, then my bands won't be thick enough and I won't be able to really see my DNA. So that segregates us into, not segregate, but like says, whatever the word is, say, whatever, moves us into gel-electrophoresis. So with the gel-electrophoresis, traditionally we're going to look at that DNA is what we're using. So we're going to, first we'll cut DNA using something called a restriction enzyme. And then you're going to insert it into these little wells, okay? And so then you turn on the electricity, which will pull the DNA from the negative end to the positive end. The reason why this works is because of the fact that DNA is negatively charged. There's a bunch of phosphate groups in DNA, and phosphate is negatively charged, which makes the whole DNA molecule negatively charged. And so it will be drawn to the positive end. So now, a biology teacher on TikTok, his name's actually a biology teacher. He had a really cool way to explain it, and I cannot remember it, but I always talk about, like, it's you, and then there is a rabbit, and then there's like, I don't know, something itty bitty, like a gerbil, right? And we're trying to race. And so you have to kind of go through a fence, right? So if you're trying to run through the fence, it's going to take you a lot longer to get through that fence than it would a snake. Thank you. It's going to take you a lot longer to get through that fence than it would for the bunny rabbit or the snake. The bunny rabbit can traditionally squeeze into the little chain link fence areas. The snake can probably just glide right underneath the fence with no trouble. So the smaller the fragment, the farther it's going to run. The farther it's going to move in that gel, okay? It moves faster. So you have the same amount of time the electricity is running, and because of the fact that those larger fragments are stuck, they don't move as far. They don't move as fast. And so that allows you to separate your strands based on size, so your largest to your smallest. And this will also tell us some information about banding patterns, okay? So if you share a banding pattern, it means that it came from that same DNA. Okay. And so this can tell us a lot of information about genetics, about who's related to who. You can do paternity tests with this because children should always have, of course, half of their parents' DNA. So you should match banding patterns with your parents, as well as you would match banding patterns with your siblings. This is also how they can test for different suspect DNA. They can do all that. And then we also have bacterial transformation. So that's transforming bacteria using external DNA. Is this a part of how forensic scientists find the criminals? Yes. So the transformation, no, the gel-electrophoresis is what helps the criminals. I know it was a delayed question. So transformation, we're picking up DNA from the environment, okay? So the way that this happens is that first, we have to take the DNA that we want and insert it into a plasmid, okay? So you will cut the plasmid and cut the gene of interest with the same restriction enzymes to provide for that sticky end that you can then use to attach it, okay? So once you get that plasmid, you're then going to add it into a solution of the bacteria and heat shock it. By heat shocking it, you're going to add it into the bacterial, so you have transformed that bacteria, okay? So in order to determine whether they've been transformed or not, we're going to grow them on different plates. Traditionally, we're going to grow them on a plate with an antibiotic in it, okay? So what this does, this is technically evolution, right? This is, we go back and forth on this. This is technically artificial selection, right? Because of the fact that I have put antibiotics into the petri dish. So I have selected for the bacteria that is resistant, okay? Yes, this is the lab we just did. And so what's going to happen is only those bacteria that have been transformed are going to grow because they will have the gene to allow them to be resistant to the bacteria. So what you have is you have a couple different features, and this question shows up on your exam traditionally. Like there's at least one of these every couple years. And so you'll have one that's LB and a minus plasmid. Minus plasmid means that it doesn't have the plasmid. So we expect a lot of growth, like a whole bunch of growth on that plate, okay? Then, yes, this is genetic engineering. Then, on the plus plasmid plate, we would also explain a whole bunch of growth because of the fact that we're doing this to ensure that these are viable, that our bacteria is still surviving. Okay. If either of these were blank, that would mean that we killed our bacteria in the process. So we would have to repeat our experiment. Okay. Down here, you see that I have the minus plasmid, meaning it does not have the plasmid, but it does have the ampicillin. Ampicillin is going to restrict that growth, okay? So I expect to find no growth on this plate because it doesn't have the plasmid, so it doesn't have the ability to resist the antibiotic, and it doesn't have any way to overcome that, okay? Then we have over here, that should be a plus, that if we have the plus plasmid, that is going to then, of course, give us colonies. Why? Because some of those bacteria have been transformed, and only the ones that have been transformed are going to be able to grow. So I hope that was helpful. So I have a bunch of FRQs that I figured I would go over just to make sure that we are fully on it. One second, there's a question in the chat. Plasmids do provide antibiotic resistance, right? I think that's why the bacteria survived in some of the petri dishes. Yes, they provide the antibiotic resistance, which will allow them to survive on those petri dishes that have the antibiotic in there. Okay, they're antibiotic resistant. That was the goal of that experiment. So we are going to do a bunch of FRQs. I'm just going to go over them. I've picked and choose different parts of the FRQ just because I know that you need certain parts maybe discussed to you or maybe reminded to you. And my students have this whole theory where they think that I'm going to give them all the FRQs. Like 4 freeze, 1 long and 3 short. Nah, not that nice. No, so my students, they have a theory. They think that, you know, they can guess my FRQs. We'll see what happens. We'll see if these are the right ones or not. Anyway, so there is something called plasmid mapping. And so there's an FRQ question from 2007. And so I think that it's important for us to understand plasmid mapping. You know, it's oftentimes an application in terms of our genetics in a way that we can use information and apply that information. Because the thing is that AP, they don't ask you straight questions. They're going to give you information and ask you to pull out that information. And then they're going to ask you to then apply that. I'm sorry, I'm still getting over my sickness. So here we have the 2007 question. Okay, so you see that we have a gel electrophoresis. And so you can see that there's been these different fragments. So what they want you to do is they want you to figure out, well, how large are these fragments? So echo is going to be 70 long. And then it's also going to have a fragment that's 30 long. Hay is going to have a 60 long, and then it's going to have a 40 long. And then if I use a double digest, basically meaning that I use both of my restriction enzymes, then I'll have a 40, a 30, a 20, and a 10. So your job is to fill in this map right here. And your job is to make it show that. Tell me where my cuts are. That would give me a fragment, 2 cuts for echo, 2 cuts for hay. That would give me a 40, a 30, a 20, and a 10. Okay. Now, it's important that we know how to do these problems. So first thing we're going to see is that I always will just add 2 arbitrary lines, okay? It does not need to be to scale, so we'll put our 2 echoes on there. Now, we know that echo has a fragment that is 30 and a fragment that is 70, okay? So we then have to think to ourselves, okay, if I have those fragments, what happens now? Well, I have a 40, a 30, a 20, and a 10 if I cut it 2 more times. So what numbers add up to give me 30? Well, 10 and 20 do. So I can cut my 30 with the hay, giving me a 10 and a 20. So that leaves me with this 70 here, and I still need to get a 40 and a 30. Well, 40 and 30 add up to give me 70. So I can cut that 70 and give me a 40 and a 30. So this is where the check comes into play. Now, this is already our answer, but we want to make sure that we do check our answer to make sure that we get everything we're supposed to. So hay should have a 60 and a 40. So you need to go from H to H and make sure that it does. So H to H, 10 plus 30 gives me 40. I'm good. And if I go from H to H, 20 plus 40 gives me 60. So I have a complete and total plasmid map. Now again, this is an application that you might potentially, definitely see tomorrow. It might not be this question. It may be number 4 on a worksheet you might have done. Who really knows what it's going to be? But you will see something like this tomorrow. So the next FRQ that we have is 2013, number 5. And so this one is looking at different species. There's 1, 2, 3, and 4. And within these species, we're going to see that we need to figure out what mutation might have taken place, right? So we have to explain what the genetic change. So if I look at species 1 and species 2, I can see that right here at the 4th position, there's been a valine that's been substituted for a lysine. So we learned earlier that that was considered a mutation. It takes such a long time for y'all to answer. So I'm just going to go ahead and say it. This would be considered a missense mutation because we have one amino acid being substituted for another amino acid. Now, the problem is that they wanted you to be very clear about this. So explain the most likely genetic change that produced the polypeptide in species 2, most likely genetic change, okay? So what you have to do is you have to say that there has been a missense mutation at the 4th position, changing the valine to a lysine. You have to be very clear because of the fact that you have it. Okay. And then when we look at species 3, you can see that we've lost these 2 right here. Okay. So that tells us that we had a nonsense mutation. So at that 9th position, there was a nonsense mutation, which led to a premature stop codon. So we have a shortened polypeptide sequence. And it says predict the effects of mutation on the structure. If we look at this one, so notice that right here, we have a frame shift mutation. And so it just talks specifically about that we need to look at structure and function. So if you have a frame shift mutation, it's most likely going to change the structure, and by changing the structure, it changes the function. The reason why it changes everything is because of the fact that you're going to have your amino acids, that R group, right, is not going to fold the same way. And because those R groups are not interacting appropriately, that's what leads to the different functions, because now it has a different structure. So now the active site's different, which then leads to different enzymatic activity. And so it's very important that we kind of make that connection. Most of the time when y'all write these answers, you're not being clear enough. You're not giving all that full information. So there is someone who's asking a question. They're a 9th grader and they're not in biology. They want to take it in 10th grade. Recommend that how many studying? I would study at least 30 minutes a night. You're good. Okay, so moving into our next question. So 2016, number 4. Okay, give me a second, y'all. Laptop died, which means I need to grab my husband's laptop so that we are ready to roll. And of course, I don't remember his password. Oh no, everything's turning off now. Because I have no power. Okay, so. No, don't close. My stupid computer. I'm sorry, y'all. Okay, so let's make this full screened. Come on, machine. Cooperate with me, please. You're really not going to cooperate? You're really not cooperating. Okay. I'm sorry, y'all. This is what happens with technology, right? Where is his flash drive? We're happy to use different light source. Okay. Yay, I have light again. Okay, so get the PowerPoint where I need to go. And the fonts can be different because it's not my computer. Okay, so back to where we were. So sorry, y'all, for my little mishap. Okay, so here we have a transcript, right? So we're looking at that we have a eukaryotic organism. Okay, you know that it's eukaryote because of the fact that we have this nucleus here with their DNA in it, allowing us to make that mRNA. That's then going to leave to then make our protein, okay? And so they're asking us that the primary transcript is 15 kilobases long, and the mature mRNA is 7 kilobases long. So it's important that we know how to apply pictures, right? So let's assume you know nothing at all. Like, you don't know anything. If you look at this picture, you can see that the white stuff is introns, and the black stuff, or shaded part, is exons, okay? And you notice from the primary transcript, that's supposedly 15, to our mature mRNA, that I go from having the white spaces to no white spaces. So logically, the introns were removed. And so you then have to say, well, where does this take place? So you see that there's that big circle surrounding it. So you can make the connection that, oh, this must be a nucleus. So it takes place in the nucleus, and we're removing those introns. Okay. The second question they're asking you is to predict the length of the mature gene if we were doing this in a prokaryote. Okay. So we talked earlier about how the prokaryotes, they don't have a nucleus, right? And they go through transcription and then translation immediately. And so they're not going to go through this RNA processing. They're not going to be cutting out their introns, okay? So it's important that you then realize, okay, well, if the primary transcript is 15 kilobases, then it's going to still be 15 kilobases when we put in a prokaryote. So that would be the answer we're looking for there. Is that it should be 15 kilobases, or you would say that it could be longer than the mRNA in eukaryotes. It's because the mRNA processing does not occur in a eukaryote. Whoa, there's a lot of stuff. The website that everything's going to be in is always apbiopenguins.weebly.com. Like, literally, go to Google and type apbiopenguins, and you'll probably find my stuff. Like, I've made a name for my name. Maybe for myself. Okay, so our next thing that we have is looking at codon charts, which codon charts are going to be all over our test tomorrow, students. Yay, codons. And so, because I can use this with our mutations, I can use this with asking you to translate something. I can ask you about different transcriptions. I can ask you what would base pair. I can do a whole bunch of stuff with this. So the question we have here is something you've seen before. This is not a new question. And so we have this gene, right? And this gene allows for a plant to grow tall, okay? And so what they want to tell you is they're saying that it encodes a certain enzyme, GA3H, for alanine at the position of 229, and the mutant allele is going to have threonine instead. So what you have to do is you have to look over here and say, okay, where is alanine and where is threonine? So alanine, I apparently didn't circle it. Alanine and threonine are right there, okay? And so you notice that all you have to do is change out any of that first nucleotides of that adenine for that guanine, and you can make the switch between the alanine and the threonine, right? So instead of a guanine, I have a adenine. So the answer we would have here would, of course, be that we have changed that codon, okay, in that sequence. Oh, I did circle it. I'm not crazy. Cool. So the next one we have is looking at this gene to mRNA to our whole big sequence, okay? And so the question asks us to do is they ask us to circle one arrow that represents transcription. So you have to think to yourself, well, where is transcription? What process happens during transcription? So transcription is when I take my DNA, my gene, and I transcribe it into mRNA. So you would have to circle one of those arrows. It doesn't matter which one you circle, but you have to circle only one arrow. If you circle both arrows, you do not get credit. It is one arrow. Be very clear in your instructions, right? Read your prompts. If it tells you to do one thing, only do one thing. So it says to circle the arrow that represents transcription. It says identify the molecule that would be absent if the enzyme is non-functional. Okay, so if this enzyme, enzyme yuck. If enzyme yuck is non-functional, what molecule would be absent? Well, enzyme yuck is going to speed up the reaction, right? Of, what is that, I3PA to IAA. So So if you don't have this enzyme, we're going to see that there's going to be an absence or a decrease in IAA. Okay, so there would be a reduction in IAA. And the justification is that we're going to see that we're not going to have that specific pathway, right? We're not going... Wait, am I on two different questions? I think I'm on two different questions. I am. I didn't give you the A. Anyways, so it says predict how the deletion of one base pair in the fourth codon in the coding region of the gene for TRPT would most likely affect the production, right? So if we have a deletion in one base pair, it's going to cause a missense mutation. It's going to have a missense mutation, which is then going to cause a mutation in that TRPT. And so since you don't have the TRPT, then of course you're going to see the reduction or the decrease in the IAA. Okay, so the same thing I was just mentioning before, but instead now we're looking at the nonsense mutation at this enzyme causing the decrease over here. And the reason why is because of the fact that we have less of the IA3PA, we are not going to be able to translate the enzyme, and then we also will not be able to make the functional protein. And then, of course, the explanation on the feedback, looking at that if a cell could prevent the production of too much IAA without limiting the I3P, right? And so if we're trying to prevent it, what you can do is talk about the negative feedback taking place, okay? So, on to our last question of the night. Last question. Yay. I feel like I've been on this forever. Okay, so we have this diagram. And of course, when you sit there and you look at this diagram, you go, oh my God, what is happening here? Miss Jones has never given me anything like this before, or my teacher's never given me anything like this before. So you have to sit there and really think about what it's showing you. Okay, so it tells you that a researcher is studying the gene expression in mice. The researcher collects samples from 6 different tissues in healthy mice and measure the amount of mRNA from the 6 genes. And the data is shown below. Okay, so this is showing you your mRNA expression. Okay, so this is where things are expressed. So you have our different tissues: liver, heart, brain, kidney, pancreas, and skeletal muscle. And it shows you our different genes. And is showing you how much of that mRNA is expressed, right? So the question that we have is, based on this information, identify the gene that's most likely to encode a protein that is essential for glycolysis. That's unit 3. So glycolysis, we know, takes place in all cells, right? So you're looking for mRNA that's going to be found in all of your cells, because all your cells undergo glycolysis. So which of these genes shows a box, like a shaded box in every single spot? Well, that would be gene G. So gene G is showing that we have mRNA in every single spot. Why? Because glycolysis takes place in every single one. Okay, so because of the fact that gene G is the only one expressed in all 6 tissues, and glycolysis occurs in all 6 tissues, or the mRNA is the only one present in all of them, and glycolysis occurs in all of them. So it's connecting back to the fact that we know about glycolysis, right? So our next question, which I apparently didn't hide the answer, is talking about that the observer notices that the tissue with a high level of gene H does not always have the gene H protein. So we're trying to figure out why, even though I have a high amount of mRNA, do I not have a lot of protein? It has to do with maybe the mRNA wasn't exported from the nucleus. Maybe we're not going to translate it because there's some type of microRNA or small interfering RNA that inhibit it. Or because I went through post-transcriptional modification. And with that, we are done. So is there any last minute questions that you have over Unit 4B? Before I sign off and say good night to you all and start grading some papers. Going once. Anybody got questions? And I know there's a lag, so going twice. Anybody have questions? And going 3 times. So, since there are no questions, I will see all of my students tomorrow. Good luck on your exam. And last but not least, remember, wait. Yes, Helena. I like how she just writes, wait. Are you sure there are no other FRQs that would show up? Oh, no, there's probably other FRQs. You know I'm not going to hand you everything. Do you have to know the backbone of RNA? The backbone of RNA is going to be the exact same as the backbone of DNA. So RNA is made up of a sugar phosphate backbone. And so you're going to see that of course, that sugar, at that 5' end of the sugar is where we see that there is a 5' end, right? And then there's also going to be the hydroxyl group. And so since you were asking about the backbone, it's literally just that sugar phosphate backbone alternating, just like what we see in DNA. So let's see, what other questions do we see? Are you sure there's no other FRQs? I'm sure that there are other FRQs. I am very mean. I don't give you everything. Any FRQ suggestions? I would know about what happens if you use restriction enzymes on things. I would know about what happens in the cell cycle. I wrote the test 2 weeks ago, so I don't remember all the FRQs that I put on it. I intentionally do this so I don't tell you anything, because you know that I don't have a very good poker face and I tell you everything. Okay, so why? Because how should we just say the Krebs cycle? The Krebs cycle's not on this test tomorrow. But you could tell us them. Dude, I don't even have the test on me. Even if I wanted to pull it up, I don't have the flash drive or anything. Let me see. What is on that test? Your long FRQ is beautiful and it pulls in pedigrees. So there's that. You have $5. Okay. Anyway, so I'm logging off. Remember, AP Bio Penguins are dressed for success. And I hope you all have a wonderful night. And yes, it is diagram heavy. Bye-bye.